the form and the function relationship between the parts and the function, but the chemistry and the structure and the function. I guess you guys did a little bit of simple and differential staining. How much theory did you guys get into when it came to why was the safranin retained with E. coli, but how come the crystal violet was what shone through in the Staph aureus? Did you guys have that conversation?
No, we just in the lab homework there were like these videos that explained it.
Okay, and so it was, here's the counter stain. It's a different color so you can see it, but it really didn't talk about how it penetrated or why it was retained.
Right.
Okay, and so that's some of the stuff that we'll get a little extra into, just so that you have the theory behind the why. Although let's face it, when you're in the moment, doing it is all that matters. And on a practical, is it pink or is it purple? That's the only thing that really matters. You're never going to be asked, peptidoglycan or lipopolysaccharide or anything like that. So let's see, let me pop this up. And I guess you guys should be able to see something that says prokaryotic profiles.
Yes.
There's some critters in there. Okay. And so I guess if we kind of just look at an overview of what we're going to talk about, these are the major parts or the major anatomy of a bacterium. The beautiful thing about a prokaryote is the pro means before and the karyote, meaning nucleus, means we don't have to worry about the command center of the cell. It also means we don't have to worry about any membrane-bound organelles. And that's probably not fair to say that. It's not that we don't have to worry about it, but they just don't bring these things along for us to talk about. And so the wall really plays a big deal here. And it's funny, if you're a plant or you're an environmental type person, cell walls belong to plants. And we really forget about, wait a minute, bacterium have cell walls. And there's a lot of other things that do too, just not the animals. The plasma membrane is here. It's exactly the same plasma membrane as you remember. So that we're not going to have to go too crazy into. Either that or it's just a refresher. You already remember it. The bacterial chromosome is very, very similar to ours, except it's smaller. And it's usually circular, so that'll kind of save us a little time. Oh, let's see, the flagella. It's the same kind of flagella. It's the same construction. It's got a same or a similar point of anchorage, but the arrangements in bacterium can be different. So we'll look at those. Ribosomes. Okay, ribosomes being sites of protein synthesis. I don't think we have to hit upon how that happens. At least we're not going to do that yet. But the ribosomes are a slightly different structure here. And really what I'm getting at is the size of the large and the small subunits. And so with bacterium, and especially with antibiotics, that size and that shape and that structure difference allows us to target the bacterial ribosomes, but leave ours alone. And so just by way of comparison, a bacterium might have a 16S small subunit, whereas we have an 18S. And so just by virtue of the 18 versus 16, the form and function difference, we can use antibiotics on bacterium and leave ourselves largely unharmed. If we tried that on a fungus, so the next time you had athlete's foot or jock itch or something like oral thrush, if we tried to use an antibiotic that was derived to target the fungal ribosome, being that fungi are also eukaryotes, we would be doing heavy damage to us. And that's why antibiotics are only a bacterial thing. We've got to use other targets for the fungi. And so kind of thinking of it from that perspective, there's a couple things that are new here. The glycocalyx or slime layer is a little new, an external covering outside of the cell wall made of carbs. Every now and again, you get some mesosomes, just incropping of the plasma membrane. It increases surfaces. Sometimes you see specialized proteins embedded here. So not that it's a special protein, but it's a special organization. If I mentioned thylakoids in a cyanobacterium or in a blue-green algae, those proteins are embedded into the membrane, the plasma membrane, but it's really in the mesosome regions where we can pack a lot of those proteins in. Other things that are interesting in here, obviously the cell wall, but there's also two structures called a pilus and a fimbriae. And back in the day, depending upon how old your book is, we didn't know about pili, we only knew about fimbriae. And so here we actually have the tools to define how they look, what they do, and how they're different. So that's kind of just the roadmap. And like I said, I'm not going to push this past an hour and 15 minutes so that you guys have the time if you want to take the test tonight, if you want to ask some questions. But this also takes some of the pressure off the calendar and the schedule, and it opens up a bunch of time for us. So it's helpful, and I thank you, and I'm happy that you're agreeable to be patient tonight, and do a light lecture so that we can kind of build a little time in. So I don't know why I start here or why we typically start with the flagella, but modal microorganisms tend to be more interesting, I guess, from the perspective of the things they can do. So to give you an example, I'm sure we cook our meat, steaks, particularly hamburgers, very carefully and thoroughly, I would think, although I'm an exception there, because we want to destroy the E. coli in the meat. Now, E. coli is not modal, which means that it's typically a surface pathogen. In other words, if you were cutting up the beef and you were a little sloppy with your knives and or you didn't clean up the carcass quite so well, any of the E. coli contamination would be on the surface of the flesh, of the meat, of the steak. And as long as you were to burn it on all the sides before you consumed it, the middle could be stone cold purple and you would be safe from E. coli. Chicken, on the other hand, we have a different pathogen we worry about. And there we worry about Salmonella typhimurium. Salmonella is modal, and so it has flagella and it has the ability to not just be on the surface, but to penetrate the tissue. And for folks who consume chicken, right, we don't want chicken a la salmonella. We don't make medium rare chicken. It's got to be cooked to 160 so that we kill the salmonella. And so just burning the surface isn't good enough. You've really got to get into it. And so that's where the flagella offers an advantage to the microbe. And as the host, we either have to phagocytize or handle the bacterium, or maybe we have to take some medication that destroys the flagella, right? That could be a strategy in terms of an agent of control. Although typically with bacterium, we don't do that. So if we look at a flagella, and I've got a couple pictures, so this will make a little sense, or maybe I'll show you the picture and I'll come back. That's essentially the organization of a flagellum when you look at the anchoring into the cell wall and the membrane of a bacterium. And so you're basically looking at three parts. You've got the filament here, which is the part that actually whips or wags. You've got the hook, so basically the transition from outside the microorganism to inside the microorganism. And then you've got some rings that anchor the rod, the actual piece that penetrates into the cell through the plasma membrane. And so these couple pieces here work to the filament for locomotion, the hook to get the transition into the body, and then the rings and the rod to anchor it into the bacterium. Basically, this is the arrangement that makes it functional. Now, flagella are definitely different than cilia. Cilia work more like an oar on a boat where it's 50% motion, 50% reset. The flagella spins 360 degrees and all of the motion translates to movement of the microbe. Now, the interesting piece of this is it really depends how the flagella spins as to whether we go straight or we zig and we zag. And generally what we're looking for is the counterclockwise versus the clockwise rotation. And I'll show you that in a second. So the other interesting thing about this is, and I don't know what folks have actually looked at over time or what microscope slides you've seen or what organisms you've observed, but generally a euglena or chlamydomonas or sperm have a flagella, right? Maybe two, but generally you're looking at one. And that's kind of what we think of as normal. But in reality, there are a lot of microbes and some really strange genera that have more than one, that have one on each end, that have a bunch on one end, that have just flagella stuck in all over the place, like they rolled on a barbershop floor. And so we can kind of look at these flagella, classify them by name, and I'll show you that slide, and make a prediction about which ones may be faster, which ones may be slower, which ones may penetrate more deeply into tissues, or is there some other type of arrangement that could give an organism an advantage, so in terms of causing infection or disease? And the answer ends up being yes, as we dig a little further into the organizations of these flagella. And so if we're thinking about words of the day, I'm almost positive it's going to be one of these, right? So when you talk about flagellar arrangements, we can talk about monotrichous flagella, So basically one on one end, like you would see in a sperm, right? Mono meaning one. You may have a critter, and that's kind of my affectionate name for the bacterium and stuff. You might have a critter that's Lophotrichus, where you have 2 or 3 or maybe even 5 flagella coming out of one end. Some of these have flagella at both ends, and those are Amphitrichus. Kind of like ambidextrous, but Amphitrichus, right? Using both, or in this case, both sides of the cell. And then the last one happens to be Peritrichus. And so these are the ones that look like they've fallen off a table onto a hairy floor. They're all over the surface. They're on the sides, they're on the top. They might be in bunches. And in general, I think we can, in fair earnest, say that the speed decreases as we move from category 1 to category 4. So monotrichous organisms would tend to swim the fastest, whereas the peritrichous ones would move forward in a much slower direction. Whereas I would say peritrichous organisms would probably spin the easiest. And so sometimes it's valuable to know that. Other times it's not. And so if we're just observing these on a slide, who cares? But if you're trying to figure out what's going to move at a faster rate, or what's going to move toward or away from stimuli and toxins faster, it's useful to know this. And so what you tend to see then is motile bacterium will move in a more straight fashion toward positive stimuli, food sources, for example, or hosts. Whereas they will tend to zig and zag, or they'll juke a predator or a negative chemical signal, right? So chemotaxis, the movement based on chemical stimuli and dangers, is alive and well here. And if you look at a couple pictures, A is monotrichous, B is Lophotrichus, and we know this because there's a bunch coming out of one end. Amphitrichus is letter C, right? So flagella on both sides. And then letter D is our peritrichous flagellar arrangement. And so A is probably going to move the fastest, then B, then C, and then D, in particular if we're thinking about straight line motions. Okay, and so kind of just straight definitions. These are some really cool electron micrographs, and you really get a good idea for how the flagella are organized and how they come out of the organisms. You also get an appreciation for the size, especially in letter C. That looks to me like maybe 2.5 microns. So you're significantly magnifying these so you can get a fairly good look. Now, in terms of movement, this is kind of interesting. What we're showing you here is that in A, on the left, with a counterclockwise movement, you're going to go forward. When it spins clockwise, it tends to tumble. Right? And that's what you're seeing here. That counterclockwise whip-like fashion tends to send an organism straight, although how straight that's going to go is anybody's guess. So counterclockwise, straight. Clockwise, we start to see a tumble or a zig and a zag or an abrupt change of direction, kind of like a wide receiver trying to avoid being tackled on game day. And so visually, I can do better than this. I can show this to you, and we can assume that when the organism is heading toward the attractant or trying to find food or a host, there is a lot more straight line activity. It's probably faster. It's probably more direct. When something is trying to avoid, you start to see a lot more tumbling, a lot more abrupt or randomness to the movement, and it makes a lot of sense. It's a lot harder to hit you or to capture you. When you change direction frequently, it's a lot easier to grab you, although much faster if you run in a straight line. So it's slower, but the zigging and the zagging usually is self-preserving. And if you guys play sports or you watch sports on the TV, right, somebody running straight gets destroyed by a linebacker or gets checked through the glass in a hockey game. Someone who's zigging and zagging and spinning all over the ice, it's really hard to catch them. All right, so this is the standard basic flagellar arrangements, how they're constructed, and typically how it affects motility. There is one other kind, and these are, I will call them specialized flagella, and they are seen in very specialized situations. And the situation that we're looking at here is, let's see, can I raise the volume? Probably. And so the situation here is going to be where we see things like, and I'm just going to raise this up. Tell me if that's any better. That should be louder now, I think. Oops, sorry. It definitely seems to be louder. Let me know if that's okay. Okay. Maybe I'll take it down a notch because people seem to be blown out of their seats. That's still good? Okay. Close that. All right. So we come down here and we see a special kind of flagella. Maybe you call them internal flagella. Maybe you call them axial filaments. And it's still motility, but notice the group of microorganisms here that we're talking about. The idea of a spirochete. Now, if we're not, there's got to be somebody familiar with spirochetes. Anybody know what exactly, or a good example of what we're talking about here? Right, so spirobacterium that are both flexible and spiral. Maybe something in the New York region, maybe something that was conjured on Plum Island could serve as a good example of a spirochete. All right, we can do it differently. Has anybody ever heard of Lyme disease?
Yes.
So that's a little better. What can folks tell me about Lyme disease?
Does it get into your nervous system?
It does. And that's where we have the greatest number of problems. So normally E. coli doesn't get into the brain. Normally Staph aureus does not penetrate the blood-brain barrier. But the spirochete that causes Lyme disease has these axial filaments. And so when you're bitten, generally you're looking for a bullseye rash. This is an arthropod vector. It's the tick. And you tend to see a bullseye rash in 4 out of 5 people, I would say. And if you find a tick embedded, normally you call the doctor and they say, is it engorged? So has it begun feeding is what they're asking. And if the answer is yes, then what they do now is they give you a high single dose of doxycycline. The jury's out, but that is the new procedure. It used to be a month of amoxicillin, which I probably would agree is too much. So you get bitten, the bacterium is transferred as part of the bite, as part of the feeding process, and it starts to move around and become numerous. Now, if you can use the antibiotics before it gets established, it's generally an easy fix. But right on, this can penetrate and influence the nervous system. And once it gets behind the blood-brain barrier, you probably don't notice anything until somebody experiences Bell's palsy. Right? And in keeping with looking at the Pfizer vaccine and the Moderna vaccine, Bell's palsy has been a condition that's happened. And what that signifies, or how you know that someone's experiencing this is literally part of the face, half of the face will go numb and just droop. The other half of the face typically stays normal, or at least the muscles you have control over. And so someone's mouth will droop, someone's face will droop, their eye will droop, and it almost has the appearance of a stroke, but it's unbeknownst to the patient, and otherwise they function fairly well. And so when that happens, obviously it's a trip to the physician. You would probably have a Western blot exam, and you probably would be diagnosed with Lyme or ehrlichiosis and babesiosis, which are pretty related syndromes. The problem there is now you're going to catch and you're going to destroy, with antibiotic therapy, the spirochetes that are circulating in the blood. But it's really hard to get all of the ones that have made it into the central nervous system. And so you'll be clear and you'll be symptom-free, but then they will come back out and you will likely see symptoms again. And for some, this is a very long process. It could go on for years or decades, unfortunately. There are some physicians now who have moved on from Lyme disease and who talk about post-Lyme syndrome. Dr. Rosenfeld's wife is one of the doctors who does those things. If anybody knows Doc Rosenfeld, might be a good resource. But it really can be a battle. And because of the nervous system involvement, thanks to these special flagella, the organism can get into the nervous system and stay there. And a lot of the times people talk about a fog, confusion, kind of disorientation. And those are the common long-term effects of established Lyme disease. Now, kind of looking at the structure, the axial filaments are really internal to the wall, but external to the plasma membrane. And they kind of wrap around the spirochete and help it to exhibit its strange, I don't want to call it a corkscrew movement, but that probably is the best way to describe it. The axial filaments or the periplasmic flagella can actually go in opposite directions and cause the organism to spin through particular tissue types, membranes, muscles, what have you, making these interesting. Now, not to downplay spirochetes as a kind of bacterium, but a lot of folks in medicine will consider these parasites, just by virtue of how they look, how they're structured, but really what they do. And I guess functionally that's right, but we still are looking at a monarin, we still are looking at a single-celled organism, so it does still fit the bacterial definition, although things could be both bacterial and parasitic, and so I think that's fair as well. Now, other things that we think about, fimbriae are a good one. And so I'm sure most folks don't think about, gee, will this organism cause disease? Should I be worried? Yeah. But a lot of organisms, if you ingest them and if they pass through the body, probably don't cause disease. And it's the ones that end up being ingested and staying, or ingested and becoming numerous, that we have to worry about. We see this kind of phenomena a lot with viruses. In terms of viruses are very specific in terms of what cells they target. So if they're not exposed to those cells or they can't find those cells, the experience that you're going to have is likely going to be mild to no symptoms. In bacterium, the fimbriae is the structure that allows the cells an advantage. And the advantage is once they touch a cell surface, the likelihood of them sticking to the cell surface is greater with the addition of fimbriae. They also may stick to themselves, right? Creating a network or creating more of a biofilm, so to speak. And whether they're sticking to cells or sticking to your intestines or sticking to surfaces, the point at which they stick then gives them the advantage to grow and use that area to become numerous. If you think of it from this perspective, and I think what you're looking in the section B is your intestines. You've got E. coli, and in your bodies, there's good E. coli, and there's other E. coli, and there's bacteroides, and a lot of strange organisms in our guts. And that's what we'd like to see. We'd like to see it be our organisms. And in looking at this picture, there is a lot of critters, and a lot of the surface area is taken up. So the more commensal flora that you have inside, the probably better protected that you are. If you ingest, let's say, a hamburger where there is E. coli H22 or E. coli 0157, so the ones that are virulent, that are able to that come with a few extra tricks, shall we say, they can stick to those E. coli that are lining the intestine, or they may stick to an open spot, and that sticking is what makes them get the advantage, become numerous, and by virtue of them growing and excreting Shiga toxin, that's really where the damage comes from, this extra ability they have. That's what causes the really nasty symptoms of 0157 poisoning or H22 poisoning. That Shiga toxin in high concentrations is what damages and shuts down the kidneys. Now, there was a question about, is a cilia and a fimbriae the same? Okay, and so a cilia is made for locomotion. Although they look very similar on the outside of a cell, cilia typically don't allow you to stick to things. Likewise, fimbriae are about attachment. They offer nothing in the way of movement for a microorganism. Aside from if this were something in the water, it would provide a little more drag to prevent it from sinking straight to the bottom. All right, but the fimbriae and the cilia, while they look similar, are definitely different, and they do two different things. Okay, so the fimbriae largely are about sticking or attaching to things. And once they attach, they could set up residence, and that's really the advantage here. Something that looks very similar to a fimbriae is called a pilus, or you will have pili. And the difference here is these are made from a specific protein called pilin. I've always said that scientists are completely unimaginative. Here's the proof. Pili are only found in gram-negative cells, so only in gram-negative bacterium. And while a pilus could allow an organism to stick, like a fimbriae would, to surfaces or other cells, what you typically see here is a pilus is like a tube that allows communication between two cells. And we're calling it conjugation, even though it's not the kind of conjugation that you would see in a protista, but it does allow for plasmids to transfer between gram-negative cells. It could also allow for messages, but generally we're thinking it's a transfer of extranuclear DNA from an F positive to an F negative cell. And when we transfer a plasmid, you're providing it with an ability. So if this is an E. coli, for example, and you eat some of these O157s, their pili could actually be used to transform some of the other cells that are in your intestines, that are in your gut. They could actually give it the genes to produce the Shiga toxin, which then means that every time they divide, you've got more and more and more and more cells that have the genes and that are likely transcribing and translating them. So the pilus here is an absolute advantage when it comes to adhesion, but also a next level advantage when it comes to horizontal or gene transfer. Now, the other interesting piece of all of this is, and this is kind of a weird thing that doesn't have to do with the slide, so I'll just stop it for a quick second. The idea of the gut or the gut flora is a strange one. And from a microbiological standpoint, your intestines are outside the body. And I know you're looking at me like I have 5 heads, but let's do this another way. If you opened your mouth and we took your intestines and stretched them out as far as we could, you know, like what, 27, 30 feet, and put a bright light at the other end, quite frankly, we'd see the light at the end of the tunnel. And so that means your mouth to your anus is open to the outside, which also means that you harbor microorganisms in there. Your heart, not open to the outside. There should be no organisms, no bacterium, no helminths, no viruses in there, viruses notwithstanding. You open up a liver or you open up a kidney, the likelihood of them being bacteria in there normally is small. And when you have a kidney infection, that's a problem. But your intestines are colonized 24/7, probably after the first 2 weeks of being born. And so microbialists look at this and say, well, that's on the outside, so it's okay. Even though you're protecting it, it's still kind of open on both ends. And so with this population that we harbor, obviously things change. You have a turnover of your microbiome, and that's to be expected as metabolism changes, as we move around the earth, as our diets change, not a problem there. But would you be surprised to know, and I'm going to go back to the slide that shows you the intestines, would you be surprised to know that some of your cells are not necessarily your own cells anymore? So if you look in here and you see there's some E. coli and you see that they're in conjunction with the intestinal microvilli, they live very close to each other. They live in proximity to one another. And so what you tend to see happening is the exchange of genetic information. And so technically, a lot of these cells, if we were to pull the DNA or we were to pull a sequence out of them, are going to be largely you, but some of the genes are going to come back to those E. coli, or some of those genes are going to come back to the organisms that are living in your gut. And so your cells are actually influenced by these. It's not just a matter of providing nutrients and metabolizing food and giving you vitamins. They actually switch some of their genetic information with your intestinal cells, and you kind of become part them, and they in turn become part you. So there's a horizontal gene transfer here between humans and microbes, which is very interesting.
I had a
Sometimes
holistic practitioner. She was all about the gut health, and I swear she changed my life. She was all about. I had severe immunity issues, and her answer to everything was, "Oh, gut health." So this totally makes sense.
it's a real thing. And the easiest example that I can ask everybody is, if New York is not your first home, and it may not be for some people, when you, I'm assuming folks have been here a while, when you go back to wherever your first home is, can you and do you drink the water? And so, you know, if maybe your first home was Mexico or Guatemala or Panama or Argentina or Haiti or Jamaica, and you've been in the United States, in New York for 20 years, when you go home or when you visit, right, you experience what most of us experience when we go on vacation. You get a traveler's diarrhea. And it's really weird because you say to yourself, wait a minute, I was born here, or I lived here for 20 years. I never had a problem. But all of a sudden, your microbiome, your gut concentration and contents are a little different. And so some people, when they go back home, have to drink bottled water. Or in the case that was just mentioned, maybe you don't feel good. Maybe you had, I'll use my wife because she's upstairs sleeping and she can't come down to yell at me. She had an exam. We went down to visit her grandmother, 2014. And we stopped at the Clara Barton rest stop, the last one, mile marker 6-7 on the turnpike. We went in and we should have known better. We really should have. We got some Burger King. That wasn't the problem. We got barbecue sauce, though, that they had packaged themselves. And that's kind of a no-no, right? Like, you don't want to do that. We're in a rush. We're tearing it south. So she eats it and starts to feel unwell and ends up with the worst case of food poisoning of all time from this Burger King restaurant. She vomited probably a good 3 or 4 days before things started to calm down. And after that, she was never the same. She developed a lot of food allergies. She developed a lot of intolerances to food. And there's a lot of things that 7, 8 years later now, she still cannot eat pre and post that experience. Further, my wife started to exhibit bodily fungi. So on her chest, under her breasts, down her abdomen, had this very strange red discoloration. And for years she said, oh, it's just sweat, you know, it'll go away. But when her doctor saw it, she said, how long have you been colonized? And she said, what do you mean? She's like, all of this funky variations in color, those are fungi. And so it changed her body chemistry to the point where she started harboring these colonial fungi in places that were never an issue before. And it all stemmed from the alteration of her gut flora. Now, nurses do have a fascination with gut flora. I don't know if it's like a support group. Yeah.
So I went to visit my grandparents in Guatemala. I forget how long ago. It was like maybe 5, 6 years, whatever. It was some time ago. Obviously, you can't drink the water there.
Well, but you live there.
No, not even them, actually, I think. But you can brush your teeth, but you just have to be very cautious about, like, if you swallow anything.
Sure.
So I remember, I think I might have maybe swallowed like 2 drops of water. I had diarrhea for about 3 or 4 days, like consistently. Like, it was like out of this world.
Yeah.
However, I don't know if there's like a correlation between it, but I suffered a lot from like really bad migraines. So right after that, I didn't really get any more migraines, like right after that whole like 3-4 day episode.
Yeah. So the link there is just purely dehydration. How much water did you really lose from 4 days of keeping nothing in and everything turning into coming straight out? And so being mildly dehydrated or severely dehydrated definitely turns your urine dark yellow, and it gives you a headache. Absolutely. So there were a group of nurses who decided to identify some people that had good gut flora. And they asked those people who they identified for good gut flora to provide samples. So you can imagine what that means. And they took this sample and they mixed them all together like soup. So basically you had a poopy soupy. They dehydrated it and they put it into capsules, like a pill.
Oh no.
Yeah. And so those became fecal transplants for people who suffered food poisonings, for people who had bad gut bacteria or bad gut health, for people who suffered some strange autoimmune or other types of diseases. And so inserted rectally, these capsules then dissolve and open, and the bacterium that were donated then become numerous and start to multiply. And doing their job correctly means they have a competitive advantage. They're going to outcompete the bad stuff you have and replace it with good stuff. And if you're into this metagenomics and this microbiome stuff, all the literature says it's important to have good gut flora. And the statistical correlations of improved health and sense of well-being were very strong. The thing that wasn't very strong was probably the desire to stick these things in your ears. Not ears, no, around the back, right? Rears. And so it's kind of sad that a technology that would be so helpful to people using basically microbes to better ourselves, if that were the mainstay, wouldn't work anymore. So would it surprise you to say or to hear that there's a company in New Jersey that's doing phase 2 trials on oral fecal transfers? Oh, those faces weren't that bad. Yeah, they really are. And it's a Bergen County company, and it's a couple German and Swedish scientists that are doing the work. And essentially, it's about coating the capsule so that it doesn't dissolve in your intestines or your mouth, and that it makes it to where it's supposed to be delivered. So if you have the time, it's a good subject. If you have strange illnesses, autoimmune stuff, it's definitely a place to look. I definitely believe the data. I think the statistics are strong. Once you get past swallowing or otherwise having this transplant procedure happen. But very, very interesting when you get down to it. Now, if a normal E. coli were to get into you, probably just becomes a part of the background. In Mario's case, in those types of areas where water is contaminated, it may have not been E. coli, it could have been Shigella dysentery. And what you were experiencing was literally dysentery. The threshold for E. coli toxicity is about 10 to the 8. The threshold for Shigella is 10 to the 1. Right? So you need 10 cells or less for Shigella. You would need almost 100 million cells for E. coli. And so that could be part of the nobody should drink this, because Shiga toxin, and I'll just type that in because it's a weird one, is very, very potent. A little bit goes a long way. So if an 0157 E. coli gets in that has the ability to produce this toxin, it could, through the pilus, give it to E. coli or other gram-negative organisms that don't have the ability. So it could transfer a plasmid, I'll put that in here too, from the first E. coli, the donator, to the receiver. And all of a sudden now, this foreign piece of DNA is in an E. coli. And unfortunately, what do we do with foreign items? Well, we try to use them. You find a cell phone, what do? You turn it on, you power it up. I find a piece of DNA, what do? I transcribe and I translate it. And so this E. coli now is then transformed and has the ability to make you sick. And in doing so, it makes not necessarily more of itself, but you spread it with reckless abandon because it's coming out of you quickly, rough, hard, probably gassy, and it's going to go everywhere. It's going to go everywhere. We're not going to ask how that exactly happened because we probably are horrified with the poop talk, but E. coli and these coliforms, that tends to be what they do. Fortunately, the glycocalyx can be a lot less heinous, a lot less nasty, and a lot less terrible, depending upon how we talk about it. So what we're really referring to when we talk about glycocalyx are capsules or slime layers, or just in general terms, molecules, typically carbohydrates, exterior to the cell wall, that some organisms have the ability to produce. Capsules tend to be very highly organized and are tight to the wall, whereas slime layers tend to be messy and loosely associated with cells. So the slime layers tend to be more benign in the respect that the cells do more about attaching and building a community when you have a slime layer. So for example, I'm sure in your bathrooms, maybe you have your own, maybe there's more than one in the house. And so, you know, the kids have one, the parents have the other, whatever the story is. You don't probably clean it every day, although some folks, absolutely they do, and good on you if you can. But you know, you brush your teeth a couple times, you spit them out in the sink, you wash your hands a bunch of times, you brush your teeth a few more times. And maybe the fifth day, you end up dropping the toothpaste cap in the sink. And when you go to pick it up, you leave a streak, or shall I say, a clean streak. And so what's happened is the sugars and the organics that you've left behind, in conjunction with the bacterium that have grown in the sink from your use, get a foothold in the sink, and they start growing and becoming abundant and producing a glycocalyx, producing an abundance of sugars. And these sugars then allow them to stick to the inside of the sink in more and more layers as it gets thicker. And then the community becomes more and more complex. So essentially you have an ecosystem growing within your sink. And a lot of the times it'll be pink, right? The film will be pink. Or maybe you see the telltale mildew where it's just black and kind of darkened. As soon as you take your finger and swipe it, you're going to know that it's a slime layer. This is the same slime layer that you probably have on the inside of all of your water pipes. And so we call that a biofilm. And this biofilm is typically not dangerous, but it could explain the differences in taste of water between your house and the neighbors' houses or the restaurants and stores nearby. You simply have different stuff growing in there. Now, I'll tell you, and you guys are missing out on watching me consume gallons of coffee in 3 hours, because if we were live, I'd be pounding it down like it was my second full-time job. My favorite Dunkin' Donuts, and I'll tell you where it was in Chester on my way to work, used to be part of the mall, and they built their own building. And I remember they closed on a Friday night and they opened in the new building on a Saturday morning. And I was Johnny on the spot on my way to Saturday class. It was actually micro, 2014. I still remember this. And the guy poured me a fresh cup of coffee out of the new machines, well, out of the machines that they moved. And don't ask me why I opened it, but I opened the lid this time. And looking inside, what I saw was offensive. So all of the biofilms in these machines dried out when they unplugged them at the old location and moved them to the new location. So when they ran those first cups of coffee, the biofilms, instead of rehydrating, were flushed out of the machines. And so every time the coffee was brewed, a little more of the flock or a little more of these sheets and these films would come off. And I got a coffee cup full of what looked like Jello or flakes swimming in my black coffee. And the guy was really cool. He's like, he apologized. I got a new coffee out of it. But it just goes to show that even in a professional setting, wherever there's water and whatever there's moisture, you're going to have organisms. And these biofilms are very classic. The other place is tidal flats or beaches. If you go out at low tide and it's been low all day, some places only get one low and one high. They don't get two. Sometimes the sands get really slippery because the biofilms tend to accumulate on the tidal flats. So that's something that you might experience. England is notorious for these. The more interesting ones, though, and the ones that generally tend to be more virulent, are the capsules. So the capsule is like a slime layer, but much, much tighter. And either of these are coded for by genetics. So there's a gene that helps an organism make these. The classic example, and I'll stick these guys' names in here too. Griffith, Avery, McCarty, and I believe it's spelled that way, and McLeod. So these are the scientists who are responsible for the principles of transformation. And Avery, among others, worked at Rutgers University. So straight in Jersey, 45 minutes from RCC. And what they did was they demonstrated the ability of microorganisms to share genes. And what they did was they used white feeder mice and they used Streptococcus pneumoniae as the organism. And strep pneumoniae comes in a couple varieties. One of them comes in smooth cells, and the smooth cells, as defined, have a capsule layer on them. Or they come in rough cells, and the rough Streptococcus pneumoniae do not have a capsule. And so the capsule makes it virulent. The capsule allows it to evade your body's immune systems. And the mouse and the human are pretty close. So while we can't give you a strep, we could give it to mice just fine. If you're thinking of an analogy, I suppose everybody's probably been to the Pearl River Parade at some point in time, or the Hoboken Parade, or maybe the big one in the city. And you might have been overzealous in celebrating, and the police may have caught you with a Budweiser or a Guinness in your hand. Amazingly enough, you look like, why did you bother me? Everybody's drinking, but everybody else keeps them in bags, right? And so that bag is kind of like the force field to the police. They see it, they might recognize it, they don't bother with it. The ones that have the label showing, those are the ones who get in trouble. And it's really like that with the capsule, right? The capsule is like the brown bag. Your immune system knows something's not right, it just can't deal with it. Whereas the label be shown, on the ones that are rough, the immune system sees everything it needs to know in order to mount a response and destroy those cells. So when we inject the rough versions into mice, they live. The immune systems beat the stuff up. When we inject the smooth, the virulent versions, the mouse drop dead hard. When we take the smooth versions, the virulent versions, and heat kill it, so basically boil it or shock it with heat, what you're doing is lysing the cells. You're destroying life function. And when you inject these pieces into the mouse or the human, it doesn't do anything. It doesn't cause disease. In fact, it may serve as a vaccination to prime your body for the next time that a pneumonial pathogen come in. So kind of like Prevnar 13 or Pneumovax 23. The breakthrough happened with Griffith, Avery, McCarty, and McLeod, where they took the dead stuff that they had heat killed, and they mixed it with the stuff that was rough, that did not cause disease to begin with. Who knows why they did this, but they mixed it up and injected that into the mousies. And wouldn't you know it, the mousies died. So they injected two things that shouldn't cause disease, and they killed the mouse. And when they did a necropsy, right, when they took blood of the dead mouse to see what was in there, they found smooth, virulent cells that they had never added. And so what happened was one of the Streptococcus gave a plasmid to the other Streptococcus. So the rough cells received a plasmid, and they began to transcribe and translate the genes for the capsule. Where did they get it from, you ask? Good question. They got it from the dead cells. You destroyed the life function. I agree. The cell was ruptured, 100% not functional. But did you remove the DNA? Did you take the proteins away? No, it's all still there. So the DNA was able to be picked up, and it was able to be utilized, hence transforming those cells and allowing them to do things that they could not do previously. Big breakthrough, a mechanism to talk about antibiotic resistance, but the capsule definitely is more of a virulence factor than a slime layer, for sure. And if you kind of look at this, where it would be cell membrane, the bottom layer here, the cell wall, and it's going to vary depending upon what you look at. And then outside of that cell wall is where you see the sugars and the protein mixes, that glycocalyx. In terms of organized versus loose and messy, the capsule is very tight and very organized. Artist rendition obviously will vary, but the slime layer really is sloppy. It's loosely associated, kind of goes off on all directions. It's also a lot less uniform than what you would see in a capsule. Just by virtue of slime layers attracting other organics, other microbes, and allowing them to live in symbiosis, which then makes the community established. It's very much an ecological succession. And that's what you see here. So in your sink, you know, you probably spit out some toothpaste and you wash your hands, and so your bodily oils coat the sink, and the organisms come from you, or flushing the toilet, or the next person that comes in. And so the substrate is really stuck to the sink. And so the cells stick to this layer. And as they grow and reproduce, they start to release their sugars. And so more and more cells stick to each other, more and more organisms can move in, more and more of these contributions can be made. And before long, you actually have a thriving, developing community. And whereas it takes trees years to do this stuff, when bacteria reproduce every 20 minutes to an hour, it's really a matter of 5 days to a week when you start to see this. All right, and just out of curiosity, however you want to indicate this, we'll do a class survey. How many folks leave their toothbrush in a holder on the counter? Is that like a big thing? Do a lot of folks do that? Yeah, there's some folks. I mean, I'm guilty of it too. I do. All right, so there's a bunch of mixed reactions in here. One of these days, for the folks who leave it out versus the folks who put it away, I guess a cap may be on it or in the cabinet, the medicine cabinet. You know, you create a lot of aerosols in the bathroom. And one of the aerosols that we create is the flushing of the toilet. And so if you make a deposit and feed the toilet and then flush, the aerosols could reach a lot of things in that bathroom, including your toothbrush. And so one of the things that folks say is, well, you know, don't keep it out because, right? Obviously, whoever comes into your bathroom, especially during the pandemic, you don't want touching your toothbrush. We know the value of not sharing toothbrushes, but now you can add a third one, and that is cross-contamination with fecal material. Now, obviously, and don't ask me how this ended up into the poopy hour. Obviously, it's your own stuff, and you know we don't want to be sharing that. But imagine now your whole family's toothbrushes are out there, and everybody's just sharing the wealth. It's one of those reasons where people start to feel really icky. Don't. You've been doing it for years. There's no disease. You're not sick. You're not unhealthy. But just think about it next time you flush, right? Think about it next time you don't clean the sink and something gets dropped into it. Makes life a little more interesting, I guess. So we've got a cell wall internal to this, and this is the last topic before we break. We're going to talk quickly about the cell walls of normal, everyday, typical bacteria. And one of the major components of this is something called peptidoglycan. Now, peptidoglycan is present in both gram-negative and gram-positives, and there may even be some in things like acid-fast organisms. It is a cool macromolecule, and it's a repeating framework of two glycan chains. One of them is N-acetylglucosamine. One of them is N-acetylmuramic acid. I'll show you the spellings because I'm not even going to try. And so we can call them NAM and NAG for short. And so the NAM and the NAG alternate. They are repeating subunits, but when properly made, they are also cross-linked by amino acid chains. And that cross-linking is what makes peptidoglycan very, very strong. And by very strong, I mean it is able to resist the body's osmotic pressures. That's what I want to say. So when you put it down and you cross-link it, it's actually strong enough to keep out your osmotic pressure, to keep water out of the cells. They don't explode, they don't blow up. Now, gram-negatives also have some peptidoglycan, but it is nowhere near as thick as a gram-positive. So in a gram-positive, I'm just going to make up a number, it could be 80 nanometers thick. Whereas in a gram-negative microorganism, it might only be 20 to 40 nanometers thick. So they're found in both, but it's a much greater percentage of a gram-positive coat than it is of a gram-negative coat. Now, the gram-negatives, because we don't need and a thin cell wall isn't helpful, a gram-negative will tend to add an external layer to the peptidoglycan, and I'll show you that in a second. It has things in it like lipopolysaccharides, so lipids and sugars specific to the bacterium woven together to make a very fatty addition to the cell wall. And so when you guys gram stain, you're applying stains in a particular order to determine, is it all peptidoglycan vis-a-vis gram-positive, or are you dealing with something that has sugars and fats like E. coli or salmonella in a gram-negative? From a nurse and a physician standpoint, this simple stain is a really good way to find out what kind of drug to give to your patient. Is it something that will pass freely through the wall because it's just peptidoglycan? Or is it something that has to have a fatty component to get through the fat in the lipopolysaccharide. And so when you look at the medicine sheets, right, there are different varieties of penicillin that one could be given, and they're tailored to gram positives or gram negatives, right? There are broad spectrums and then very narrow spectrums, depending upon what these walls are made of. If you were to look at peptidoglycan, these are the actual chemical molecules, right? N-acetylmuramic acid and acetylglucosamine, and they are cross-linked, so sheet to sheet, molecule to molecule, with this short layer of peptides, alanines, glutamates, lysines, and linking the two is a glycine bridge. And so this allows it to be strong. And I don't know if you remember what glycine looks like from chemistry, but also very flexible. Now, for those who are into drugs, and by that I mean chemotherapeutics that work against microbes, although why leave out the rest of the stuff? Do the rings in this slide look like anything to you? Right? Does it look like any kinds of medications or drugs that you've seen pictures of? And the funny thing about it is, it's probably a very general and a very loaded question. But the hint here is the similarity of amoxicillin or penicillin to peptidoglycan is actually kind of strong. And what I'm getting at is the beta-lactam ring in the penicillins looks very much like the rings that you're seeing in front of you. And that's where the strategy of taking penicillin or amoxicillin comes in. Because if you're a bacterium, it's expensive to make peptidoglycan. But if you see something that has a ring that's very similar to what you see in the bottom right picture here, and if you see a lot of something with a ring in the environment that you could use and turn into peptidoglycan, I would probably be lazy and save my energy for other things and incorporate the drug that has a ring that's similar to it into my physiology. And unfortunately, penicillin does that. Fortunately for us, it doesn't cross-link. And so when they do that, you make them susceptible to osmotic pressures. And so you stop their ability to reproduce. Your body actually helps destroy the microbes by flooding the cells and blowing them up. And in their reduced or static number, your immune system takes care of the rest. So it actually is an ingenious strategy that we learned the mechanism after we observed the phenomena. So that's actually pretty cool. And things like amoxicillin and vancomycin all have this ring, although they're organized with side chains that look different. So you've got your generational antibiotics, first gen penicillin, second gen, third gen methicillin, fourth gen vancomycin, and so forth. So the ring is conserved, but the rest of the stuff changes, and it's actually a good strategy. So we typically can group cells based on their 4 types of compositions: gram negatives, gram positives, bacteria without cell walls, so stuff like mycoplasma would count, generally not infectious unless you have a compromised immune system. And then things like tuberculosis or mycobacterium go under the 4th category, where we say bacteria with chemically unique walls. The uniqueness when it comes to tuberculosis is actually the mycolic acid in the wall. And that's what makes gram stains ineffective with these mycolic acids, and why we go over to acid-fast staining. So those are our basic 4 groups. Here is the difference, or at least visually, between what gram positive and gram negative looks like. So on the left, you've got your gram positive organisms. On the right are gram negatives. Both of these have a phospholipid bilayer, so no problem there. Both of these have cross-links that go through their cell walls. No problem there. The gram positives have a very thick peptidoglycan layer, no lipopolysaccharides, and if teichoic acid or lipoteichoic acid or whatever cross-linking the layers of peptidoglycan. So a very thick and a very strong layer. On the right side, we've got lipids again, almost another phospholipid bilayer infiltrated with polysaccharides. So it really is a sugar and a fat layer. and they've got porins or transporters or membranes where drugs or other types of substances could pass if they were the right affinity, the right charge, and the right size, right? So really two separate layers to deal with on the gram negatives, whereas on the gram positives, it's just a fat layer of peptidoglycan. Now, the chart that comes after this will explain gram staining, but let's look at the organisms and let's talk about what you did. All right, how about you tell me how you gram stain? And let's walk through what happens. Anybody remember from the lab last night or 2 nights ago? So you have your slide and I want to gram stain this. What's the first thing I should do? All right, so we're going to apply crystal violet. And typically what we would do is called flooding the slide. We would pour a layer over the entire thing. And we let it sit about a minute. And what's happening is it's imparting color to everything. So it soaks into the peptidoglycan and it actually gets onto the lipopolysaccharides. Yeah, we would heat fix it before we started the staining. Absolutely, we would need to do that. Because then when I flooded it with crystal violet, if I didn't heat fix it, I would wash off all my bacteria. That would be worthless too. So we heat fix after it dries, then the crystal violet. Spot on, spot on. Okay, after the crystal violet, what happens? All right, so we add the iodine, and that's exactly what it does. It kills and enhances the primary stain. And it is also a mordant. So the mordant is just a killing agent. All right. And then we got to rinse. Absolutely. After about 60 seconds, we rinse. And then the alcohol. Okay. So if someone were ever to ask you the most critical step in a gram stain, the alcohol is by far the most critical step. Because when you hit the alcohol on these cells, if they're gram positive, you're dehydrating the pores in the peptidoglycan and you're shutting them. If you add alcohol to a gram negative, it's actually solubilizing the sugar and fat layer and washing it away. That's another reason why the alcohol is sometimes called the decolorizer, because it's going to remove the primary stain from the negatives and it's going to seal the primary stain into the positives. After the alcohol, and let me tell you, it says a drop and rinse quickly. It really is a drop. And again, I'm sorry you guys can't do this in the lab. It is as much of an art as it is a science doing this. And that's what makes the staining kind of fun. That and invariably each of us go home tie-dyed. And so now in the gram positives, we've got a dark purple color on the cells, dark purple color in the peptidoglycan, and we sealed the pores shut. So the counter stain, which I'm sure you guys remember is safranin, may not penetrate, but certainly will not be seen over the purple in a gram positive cell. In the gram negatives, though, the peptidoglycan might be exposed. And so the safranin soaks into the peptidoglycan that's now been newly exposed, and it shows pink. And so if you over decolorize, this is why it's the most important step, you could actually turn a gram positive, gram negative, by washing away all of the peptidoglycan and all of the crystal violet inside it. So that's where that step becomes most important. And if we summarize what we just kind of talked about, the crystal violet, step 1, after you've made the slide, allowed it to dry, and heat fixed it. Spot on, I don't want to forget that. The crystal violet imparts color to everything, absolutely what it does. The grams iodine is a mordant, so it is a killing agent. It also makes a CVII, so a crystal violet iodine complex. To the naked eye, it looks like the muddiest brown, I won't say it, we've been there too many times, the muddiest brown color, but at the microscopic level, it makes the purple pop. The alcohol then dehydrates, so in the case of the gram positives, it seals it in. In the case of the gram negatives, it decolorizes. It takes all of the fats off and the crystal violet that's bound to it, and you get a decolorized cell with some peptidoglycan out there. Then you hit it with the counter stain. It's not going to penetrate the peptidoglycan and the closed dehydrated peptidoglycan in the gram positive cell. It'll stay on the surface, but it won't matter. You're not going to see it over the purple. And in the gram-negative, and you're going to leave this for 5 minutes, by the way, it penetrates the exposed peptidoglycan, and then you rinse it off and look at it, and you'll see that lovely red color where they're negative, or that lovely purple color in the case of where we still have positives. All right, and so that's probably a good place to stop. That's enough technical speak, and I'm going to hit the stop recording button here.